Okay, a few questions from online. First, so for Steve, the question is, do you have any data on the rates of the mutations across different ethnicities?
Yeah, very, very interesting question. So it's, you know, you'll be surprised to know it's complicated. So it turns out that the rate of Dectin-1 mutations in the Y238 Dectin-1 mutation in Asians in the general population is vanishingly rare. And yet 13 of the 14 Asians that had disseminated disease in our cohort had Dectin-1 mutations. And Amy and I were just going over that this week to think about whether we should go back and see if that's on a particular haplotype. Is that a founder somewhere that we haven't recognized? So the Asians in our group had the Dectin-1 mutations. The Caucasians had the PLC gamma-2 mutations, which is not surprising. That's where it's been described as a cause of inflammatory bowel disease. The more interesting one was really in the DUOX and the DUOX-A1. And each one of those DUOX and DUOX-A1 mutations were distinct, and or almost all of them were distinct mutations that were loss of function. And so that was seen almost exclusively in our African American population. So it's sort of interesting. We've got three ethnic groups, three very different sets of mutations. And we're now trying to go back and figure out why is that? Why are those things what we see in those groups?
Thank you. The next question is for Anita. We know that different fungal isolates, in parentheses, strains, have varying levels of virulence in a mouse model. Could you comment on regulation of virulence in a single strain? Specifically, have you studied the effect of serial in vitro passage on altering or diminishing virulence?
So I'm sorry, I'm not sure I understood the question. So I'm not sure if the question is whether it's useful to look at virulence in a single strain, which may or may not be generally applicable, or whether it's essentially a commentary that looking at virulence in a single strain may not be sufficient, and it will be interesting to see whether that holds true, because there certainly will be, I'm sure, multiple virulence strategies that differ, say, between Posidacea and Imidis, as Bridget was pointing out. You know, there are already clear phenotypic differences, and there are many phenotypes that we haven't really exhaustively interrogated yet. So I'm sorry, Mark, I'm not sure if I'm answering the question.
I'm not sure I can clarify that, but in the field of fungal plant pathology, passaging strains outside of a host often results in reduced virulence, so maybe they're thinking about something along those lines.
That absolutely makes sense. And I mean, we do have, thanks to Bridget and others, to John and many individuals, there are collections of clinical isolates, not all of which have been passaged extensively. And so I think looking at the biology of those is going to be important. And certainly we and others are trying to do genome-wide association studies with some of those collections to try to identify loci that are relevant to virulence. So those are going to be important complementary experiments to studying lab strains for sure.
So it seems like maybe they were asking whether you had tried serial passage to identify reduced virulence strains, which might help identify transcription factors important for virulence.
We have not, but I don't know if others have. We have not tried that yet.
No, we haven't done that either.
I don't think anyone has. Great idea.
The next question, also for you, Anita, is that it doesn't look like the arthrochonidae are being phagocytosed by the macrophages. Do you see phagocytosis, then spherulation and escape, or just contact then spherulation, or something else?
We see phagocytosis. So we can, I didn't show you those data, but we can do inside-outside staining, and we see not as good not as efficient phagocytosis as we do with some other fungal pathogens like histoplasma. And right now, I'm of course forgetting the exact kinetics of it, but it is reasonably efficient. The majority of the arthrochonidia are phagocytosed. Of course, that's somewhat MOI dependent. You can kind of swamp it out if you put in a lot of arthrochonidia in there. And then we kind of see this mysterious thing where the spherules appear. So we do see some spherules that appear to be intracellular when they're smaller, and then we see the appearance of extracellular larger spherules. So the presumption is that there's, and we see that the macrophages are lysing because there's release of cytosolic enzymes from the macrophages. So the simplest explanation would be that there is some kind of escape of intracellular spherules to the outside of the macrophage, but we really haven't documented that precisely yet.
Questions from the audience? John?
This is for Anita. In the the study you just talked about with the macrophages and the arthrochonidia, does temperature and the amount of CO2 matter in making the spherule, the arthrochonidium to spherule transition, or is it just the macrophages?
So what I showed, so if you keep the conditions identical but take out the macrophages, you get robust hyphal formation. That was that first video. And so the conditions under which you get spherulation with the macrophage are the conditions that are optimal for the macrophage. So the 37 degrees, 5% CO2. So under those conditions, without the macrophages, you get robust hyphal formation, and with the macrophages, you get robust spherulation.
Got it.
Anna?
Yeah, Anna Litvintseva, CDC. Also, question for Anita. So really cool movies. I haven't seen those before. So has it been tried with other cell types? Is it always macrophages or?
We haven't tried with other cell types. John, I don't know if you want to comment because some of your previous work is relevant there.
Sure, be happy to. John Galgiani, University of Arizona. Actually, the work that I did in the first 18 months after moving to Tucson was based on studies done by Abe Browdy in 1956. And he was the one who first showed the transformation of arthrochonidia spherules in contact with a buffy coat preparation. And we found the same thing happened with mononuclears or with neutrophils. But the other point was that subsequently Steve Klotz and David Drutz put together impermeable capsules where he put arthrochonidia inside the capsules and then surgically put them subcutaneously in rabbits and was able to demonstrate the arthrochonidia in this cell-free milieu inside these capsules would undergo transformation of spherules. So that's where, maybe, John, that's where you're coming from with the CO2 question. And they postulated it may be not so much the cell contact, but other factors.
Yeah, I mean, it makes sense that that would be a much more complex milieu than what we're doing in terms of simply just adding the arthrochonidia to the macrophages. So it makes sense that some of the parameters, like whether contact is required or not, would be different.
In the back there?
Hi, this is Manish from UCLA again. This is a question for Steve. Steve, great paper. And congrats to Amy on this work. The questions about the Dectin variant, the loss of function variant that you said was pretty common. Do you think it then affects those patients who get cocci infection and go on to dissemination, or control different? Does it not control that transition from infection to dissemination? I'm always thinking about the severity side of infection. Because, as you mentioned, 100,000 people are getting infected, but only a portion are having the severe outcomes. And maybe as a follow-on, the TNF aspect that you pointed out for all of these do TNF inhibitors increase the risk of disseminated cocci? I'm sorry, I should know that, but I
Yeah.
actually combined
Yeah,
question for you.
yes, and if you follow enough GI patients, you will know. It definitely increases the risk, and that's one of the reasons that we thought that the TNF was a very likely biologic explanation. There, of course, might be others, but the risk for people on TNF inhibitors is substantial. There have been calculations made of it, and I don't remember the number. Yeah, I think that the dectin mutations are a marker for failure to control, so I think what's fascinating about Anita's work is at exactly what level is that failure to control? That is it occurring at the level of autonomous TNF production? Is it really T-cell mediated TNF production? Is it macrophage mediated T-cell production? Who's behind it? At what point does it contribute? And I think the next set of experiments we'd like to focus on is trying to figure out exactly where in the mouse model does dectin-1 deficiency lead to escape of cocci from control. I think that's a critical question, Manish, and I wish that we had a better answer for it. But yes, TNF's important. Yes, I think that the dectin mutation is a predisposing factor for severe disseminated disease, more so than for local controlled disease.
You?
So I have two questions. The first one is for Steve. So it's very nice to see your data demonstrate what could be the protected immunities. And look like dectin-1 does hit on the innate immune response. And intuitively, we would think that the T-cell response, the adaptive arm, will be affected. And I just wondering whether you have any evidence.
So I'm sorry, is the question whether T-cell function is affected by these dectin mutations?
Yes.
I think the answer to that will be yes. Now, I'm not the right one to answer that in depth, but the paper from Mihalis Leonakis's group, at the same time that we were doing this work on disseminated cocci and these dectin mutations, Mihalis was characterizing a patient that I had seen almost 20 years ago, who had biallelic dectin mutations, who wound up with disseminated infection with Caseocola, Corinospora caseocola, the cause of cucumber rot. And it's clear that there are both myeloid and lymphoid effects of mutations in dectin-1 that we can demonstrate in the mouse model. In the human model, I think what's important is what you see in that the transplant experience in aspergillosis, that there are both hematopoietic and somatic features to dectin-1 deficiency. And this makes me think that, some of the signaling that might be going from dectin through PLC gamma to DUOX in the epithelium could be really what is part of the somatic component that you see in the recipients who have these problems with the dectin mutations. So yes, I think there is a T-cell component to it. No, I'm not sure how much more prominent that is than the macrophage or alveolar macrophage or even epithelial component. But I think Mihalis Leonakis would be a much better person to answer that question, and his paper might address it, and I've forgotten it.
Yeah, I would like to see it, and because we demonstrate that in mice, and I just wonder what happened in humans. Well, my second question is addressed
to... We're going to have to take a break now because we're a little bit behind, so please bring your question down front, and everybody, it's a short break, so we want to reassemble at 4:45 to get the next session going.